In this 15 minutes, I would like to focus on what we are studying and what our lab is interested in terms of cellular senescence and senescent cells, which became in the last decade a very interesting topic in the longevity and aging field. So just, I'll use this. As you all know, with aging, there is increased probability of different age-related diseases. Let's see here. Here, yes. And there is a notion that there are common molecular and cellular mechanisms that underlie these diseases, and these mechanisms summarized, I think, in the literature in several reviews, and there is a picture from one of them, and this mechanism are called hallmarks of aging, and cellular senescence here is one of the hallmarks mentioned here. So why cellular senescence is a hallmark of aging, and what does it mean? So there are senescent cells that are formed in response to stress, and we will see how in a second, but there are cells that are formed also in the young organism, but the immune system can take care of the cells and resolve their presence and to maintain healthy turnover of senescent cells. But in the older organisms, there is not only increased formation of senescent cells because internal type of stress and all kind of external types of stress and damage that we experience during our lifetime, but also reduced immune surveillance of senescent cells leading to their accumulation, and the accumulation of senescent cells leads to chronic inflammation in their microenvironment and promotes age-related diseases and conditions that are listed here and many others in their microenvironment. So this is why senescent cells are, when they accumulate with aging, they are damaging and they are pro-aging, and there are studies showing that elimination of these cells extends health span and lifespan in different models, mainly a mouse model. So how is possible that cells that are damaging to our body actually accumulate in the body? Why? Is there something in nature, something damaging can be eliminated, right? But probably there are things that senescent cells do that are beneficial, and this is why they are necessary to form, and only when they accumulate with time, they execute their deleterious effects. So senescent cells form in response to various types of stress, and these types of stress activate molecular pathways that lead to short-term responses which are essential. And these short-term responses are tumor suppression, which is essential through lifetime, limit tissue damage, and also participate in embryonic development, mainly in the transient structures. So senescent cells are necessary, but then when they accumulate with age, they secrete, as I mentioned, pro-inflammatory cytokine called SASP, senescence-associated secretory phenotype, that can promote tumorigenesis and tissue aging in their environment. So the immune system tries to uncouple this positive, necessary short-term responses from the deleterious long-term responses. And over the time, we and many others thought that we can probably mimic immune system or help the immune system and eliminate senescent cells directly. And then we have discovered a few of these pathways over the years, and some of them give us the possibility to use known molecules in order to eliminate senescent cells in our mouse models, and ask questions, indeed, how accumulation of senescent cells leads to tumorigenesis, or how it affects tumorigenesis or aging. So just a few examples of this study. So one study where we wanted to know the effect of senescent cells in early lesions of cancer on tumorigenesis later on, and we all know that from quite many studies that these premalignant lesions accumulate over time and in mouse models and also in humans, and they are not detected because they don't do anything and it's not possible to detect them. But some of them with time wake up and become tumors. So there is a long-known mouse model and a model of role of senescent cells in tumor development that is summarized in 2005, 20 years ago. But the idea is that there is an oncogenic mutation in some of the cells, and these cells can proliferate, but then they become senescent and form what's called premalignant lesion, but then one or more cells can escape these lesions and become a tumor. So, okay, we have a stage where there's a lot of senescent cells, and this is a long stage. So we took a mouse model that goes slowly through all these stages over many months in mice, and we have a tool that I mentioned that we can eliminate senescent cells only in these early lesions, and then ask more than half a year later, after we finish the treatment, half a year later after we finish the treatment, if the tumors will develop. And indeed, we have seen that when we eliminate senescent cells from premalignant lesions, we have a more than 75% reduction in tumor formation in mice, and this was in a model of pancreatic cancer. And, it was published a few years ago and was done with, in collaboration with great friends from the Hebrew University of Jerusalem, Dasa Medical School, Itai, Val, and Gidi. So this was about cancer. So what's about aging? Can we use the same tool to ask what is the effect of senescent cells during aging, especially in the mouse models where senescent cells accumulation is increased due to the decreased immune surveillance? So we had one of these mouse models, and what we have found that normally there is accumulation of senescent cells, which is under the control of the immune system, and then if we remove this component, there is accelerated accumulation of senescent cells and increased risk of development of different age-related pathologies in mice and shorter health span and lifespan. And we can treat and eliminate senescent cells and extend median lifespan in mice. And in the same study, we have shown that we can see the signatures of the expression signatures on the molecular level in the treated mice showed a more younger expression profiles than the old mice. So this also was published already quite a few years ago. And now for the more recent studies, so that we can identify senescent cells by different phenotypes, and we try to use more and more markers to identify senescent cells more specifically. And in the senescent field, there is a big kind of discussion. Can we identify senescent cells by the single marker? Probably no, by many markers. But usually we would like that these markers would reflect different sides of senescent phenotype that are listed here. And then we use this approach, looking by many markers using the technologies that's called CyTOF, to study senescent cells in different pathologies. And in one of the studies, in collaboration with Michal Schwartz from our institute, we looked at a model of Alzheimer's disease where we could eliminate senescent cells now in a model that was just mentioned in the previous talk, 5XFAD model. And for more details, you could visit a poster by Noah Rahmian, who was the first author on this interesting study published this year. So, but just to summarize quickly, old mice and mice of this Alzheimer's disease model and one other Alzheimer's disease model accumulate senescent microglia cells, which we can identify by signature of multiple proteins. That markers of senescence, they correlate with markers of TREM2 and ApoE, that ones that are important for the interaction with the immune system. And then we can use the senolytic approach to eliminate senescent cells in the body. And this lead to improved, let's say, kind of memory test, this novel object recognition test that is depicted here when mouse kind of should remember if it have seen an object or it have not seen an object. And in this, these mice, they fail in this test, but when they are treated with the senolytic compound, they perform much better, suggesting that elimination of senescent microglia cells improves at least some functions in this Alzheimer's disease model mice. So let's get back to this kind of schematic presentation of different diseases and conditions in humans where senescent cells play a different role. And we know that lung diseases are, chronic lung diseases are among the more prevalent age-related diseases in humans, including IPF, COPD, and so on. And chronic lung inflammation happens many times. So we started to look at these models, again, with the same kind of techniques, identifying of senescent cells, by a combination of different markers. And when we looked at this, also with bioinformatic help from Avi, from Uri's lab, we have identified that senescent cells, specifically P16 positive cells, upregulate immunoregulatory marker, very famous marker called PD-L1, that senescent cells of different types upregulate PD-L1 in correlation with one of the major senescence markers, P16. We identified that P16 actually molecularly regulates protein stability of PD-L1, and this is where it's normal, say, well-known molecular pathways through regulation of activity of CDK4, and that we can use activating antibody anti-PD-L1 to eliminate senescent cells. But one kind of important notion from this study is that if we just were looking at expression of PD-L1 on the Y and P16 on the X, and we can see that there is a correlation between P16 and PD-L1, but we can see old in red and young in blue, and we can see that regardless of P16, the level of PD-L1 in the old mice is way higher than in the young mice. And this is in different cell types. This is the epithelium here, just to think how much levels of this immunoinhibitory molecules are increased with aging. So time is short, and let's summarize what we know about senescent cells and what we think now about senescent cells. So senescent cells are normally formed in response to various types of stress, and this is a normal and necessary mechanism, and immune system keeps senescent cells in check by maintaining normal turnover of senescent cells, and this turnover gets disrupted with age. Senescent cells accumulate, they inhibit their own removal by their accumulation, they lead to local inflammation, to tissue dysfunction in their environment, and they promote different age-related diseases. So so we have seen in this recent study that I just mentioned that PD-L1 is one of the factors that inhibits the turnover of senescent cells in the old mice. So just thinking about senescence in aging and different types that we already heard, and we will probably hear here about life extension, and we all know that started with this, that there is increase in incidence of different age-related diseases with age, and this is kind of would be a 6-span, and then the time before these diseases appear would be the health span. And then if we would like to extend lifespan, there are more than one possibility how we would extend it. And what we really would like to achieve is this increase in health span, where we make the sick span as short as possible and the health span as long as possible, and not just extend the lifespan where we actually extending the sick span and the extending kind of suffering. It's not what we just like to achieve. So just lifespan might not be the only factor that we need to think about in longevity research. With this, I would like to thank all the collaborators, all lab members, and funding sources that help us to do our work. Thank you very much.
Questions for Valery?
Is there any preference about the tissue type where they accumulate, or if they accumulate in a certain tissue, would you see it also in other tissue?
Can you repeat the question?
Yes. So the question was, are senescent cells accumulate in certain types of tissue, and does it affect the tissues in the surrounding? So we are actively studying this question right now, and we and actually many other labs around the world. So senescent cells can appear in multiple tissues and multiple cell types, especially cell types that normally proliferate, and there are many such cell types in our organism in different tissues. And they definitely, the idea is that they would affect probably the senescence in their environment and in the neighboring cells and in this, let's say, in the same organ.
More questions? I have a question. How does it work when you eliminate senescent cells into precancerous lesions? There's less cancer later. How does that work, do you think?
So one of the major drivers of cancer in many types of cancer is chronic inflammation. So while we eliminate senescent cells, and there is a big accumulation of senescent cells in the pancreas in this model, we reduce the amount of inflammation for a long period of time, and by reducing the amount of inflammation, I think we're reducing the cancer incidence because inflammation is tumor promoting. Okay, thank you.
Thank you very much.